See if Chris has what he needs. Can you see at least one of the slides? Okay, that's great. Welcome, everybody. And there are carrot cakes here. You don't have to sit in those 3 seats in order to justify carrot cake. Happy New Year. So, why Unbridled Passions? Well, bringing local folks in for an afternoon talk is not so easy. So you have to over-advertise a little bit. But actually, it's true. One of our concerns is that our patients have sort of a very passionate motivational system that's not very well regulated. And in fact, that's one of the core features of relapse vulnerability. I've got this sort of on my, tell me if this is loud enough for you guys. Okay? All right. This is, as you know, not a solitary adventure. If you've done brain imaging, you know it takes a village. On this slide are a group of people who have contributed to the work that I'm going to show you, and the names in yellow are still active. There are folks who've actually contributed who are the white names, who have gone on to other positions, other things. We're grateful because without all of these people, and I will not introduce them all because then we'll be finished with the 20 minutes, this kind of work could not happen. But it's a multidisciplinary team, so I'm an investigator and I have grants, but without physicians, nurses, clinicians, research assistants in the trenches, postdoctoral fellows with new hot ideas, I would be but a mere cog in the wheel. We also have a handful of ongoing collaborations with people inside and outside the university. So 2 of those are close by, over on the far right of the slide. We have an ongoing collaboration looking at cues in eating disorders with Dr. Michael Lowe's group. This is actually from Drexel. We had an R1 together. And for sexual risk behaviors with Ann Titleman, who's in the School of Nursing. And Paul's been leading a lot of those analyses. In addition, we have some things that are going on with people that are often here, not here at the moment. Rick Christ is actually sort of our genetics analysis person, and we'll show a couple of examples of the way that we've been examining genetic variation in people's vulnerability. We also have high hopes of bringing PET tools to bear on these kinds of questions. And we've had an ongoing collaboration with Bob Mack and his group in that regard. We have a recent collaboration with Mike Saladin at MUSC, who hopes that using our cocaine cues, stirring up the brain, and then disrupting reconsolidation of the memories about cocaine might be an intervention. And so he's actually working with our cues doing that. We have a paper that was just out this week in collaboration with Corinne Weers and some of the folks at NIDA who were interested in, as we have been, motivation that begins outside awareness, unconscious motivation, with the cues as a trigger. And we have a collaboration that Paul's leading with Vanessa Troiana at Geisinger looking at orbitofrontal morphology in our cocaine patients. So those are things that further enrich the regular stream of funding and the activities that we do, which are, of course, focused on the general topic of addiction. And for our lab especially, we're interested in the issue of relapse. This is the return to drug use after a period of being drug-free. It is the clinical problem for treatment populations. They go through a treatment episode, they think things are going to be better, and then inexorably, they often relapse. For us, though we recognize that there are a lot of different cues, crimes, stresses, and even withdrawal that can tilt scales in the direction of relapse, we've been very focused on the role of cues, and I'll talk more about that. How shall I talk about cues? So you can either look at the carrot cake in front of you, or consider a French toast that you may have encountered in the past. Thinking about the Grand Marnier whipped cream, how it feels in the back of your throat, the warm maple syrup, the taste of the strawberries against the toast. If you were thinking about that in a very vivid way, you might feel a little pull, sort of a go moment. Or maybe you're not a strawberry person. Maybe it takes chocolate to do it. So let's think about chocolate stuffed French toast with a little bit of ganache, again some whipped cream. And if you really still aren't there yet, what about combining it? So that we put strawberries and whipped cream and bananas and chocolate. So if you are feeling a little bit of pull, a little bit of go, you don't have to embarrass yourself by saying so, but you're really just a member of the human race. We Humans are exquisite reward detectors. Part of our evolutionary legacy is an ancient reward circuitry that actually helps us appreciate rewards like sex to keep the species going, and food to stay alive another day and have more sex. So we basically are always appreciating rewards and their signals. That's part of what we do. It's really important. And it sounds like a very innocent thing, and more is better, right? So in terms of survival fitness, individuals who had a very strong reactivity, and maybe they didn't have chocolate mousse at that time, maybe they did have profiteroles, I don't know, but basically would have been having an edge in terms of survival, right? Because they were the ones who were the earliest bird for the worm, and responding to the signals for reward. A very useful, adaptive thing. There's an interesting potential dark side to this. What happens if you have a lot of appreciation of rewards, but now you're in an environment where situations change? You have your same old brain's interested in rewards. Now you have unlimited access to, for example, calories. Or, in the case of drugs of abuse, things that really activate that circuitry in a supranormal, supraphysiological way. We think it could be potentially a problem, such that this pull toward rewards could actually be a potential relapse vulnerability. If you have a system that's exquisitely sensitive, very responsive, say hyper-responsive, even though evolutionarily you might be the fittest of the fit, it could put you potentially at a disadvantage. So these first few slides are just context about the way that we've approached this question, and the last 10 minutes are samples of data that talked about the way we've actually collected data on the topic. So this reward sensitivity is something we've been fixated on. We know that that's not the only system in the brain, and I won't spend much time on it today because of the limited time, but the reward system, the GO circuitry, holds hands with these upstream circuits, the so-called stop circuits, the executive, top-down control centers. And those are complementary systems, and they actually can be complementary vulnerabilities. And compromises in the frontal circuitry also being very important, because if you don't have very good brakes, that can be an issue. So we think that the delicate balance between these systems is actually very important for our everyday behavior and for understanding some of the vulnerabilities I'm going to talk about. So very quickly, in a cartoon way, broad brush, in a normal adult brain, you've got this frontal circuitry, we'll call it the stop circuitry, that's responsible for modulating these downstream, throbbing, pulsing GO circuits, so that, hopefully, if things go well, that you are able to think about the consequences of acting on the impulse toward reward. Thinking it through. You were pulled, but you have a circuitry that's saying, well, maybe not. Maybe not the night before the test. Maybe not at the office party. Giving you some braking power so that your impulses toward reward are managed, and that for the most part, though we stumble along and we fail from time to time, you generally are making decisions that are advantageous for you. But what if you have a brain that might be vulnerable, in which circuitry, that's just to wake you up, just in case you don't. Either is not as well connected, or that one part or the other is not as well developed, not communicating. Then you can have a system where basically there's poor decision-making going on, because now you may have this sort of rogue GO system, not enough top-down modulation. And you could have poor decisions, taking risks, and it could be that this kind of imbalance is something that could not only set you up for relapse, it's the kind of thing that could potentially set you up for addiction itself, right, in terms of basically these same circuits. So in the very broad brush way, two-box models, we think about this balance between GO and stop circuitry, and I'm going to talk today about some of the examples about the way that we've focused on the GO side of the motivational versus regulatory coin. And we were struck very early on by patients' descriptions of how this circuit could get activated. In the early 80s, when I was yet a child, the cocaine epidemic was upon us, and you would have patients telling you about seeing things that reminded them of their favorite drug. We were at 35 at the time, the VA was only a few blocks away, and there would be sort of like a Hansel Gretel stream of little bags, little zip bags or little glassine bags that would have either crack pellets in them or other paraphernalia. And they would describe that when they saw something that was very cocaine related, that had been a good reliable signal for the arrival of cocaine in their usual day-to-day drug use, that they would experience a wave of sensation that they gave very colorful terms to. They would talk about basically a wave that starts in their toes and comes up and causes their stomach to go flip and their heart to go pat and their ears to go hyperacute, head tingling and light. They could almost taste cocaine even though they hadn't used it, so it was almost like a sensory hallucination. They also felt a little tinge of euphoria though they hadn't used it. So all of this was pretty impressive when we heard about it. Sorry, wrong direction here. I didn't mean to touch that. I did. And basically, those descriptions caught our attention, and we wondered if there was a way that we could capture that state in the sterile laboratory. Because that's something that happens out there where there are really good reminders of cocaine. None of us are really good reminders of cocaine. Prepare yourself for the vulnerability. So rather than saying throbbing, pulsing desire states, which is a real mouthful, I'm just going to call these ghost states, and say that basically the very simple idea here is that this is good old classical conditioning, right? As with your cat approaching the can opener when it's mealtime, things that have associated with the arrival of the drug have acquired all kinds of interesting abilities to pull out motivation and sometimes some interesting physiological symptoms, subjective symptoms. Good classical conditioning. So we thought if we could actually essentially recreate that situation in the laboratory, maybe we could see this state in its rogue, unmodulated way, and basically see if we can modulate it with behavioral therapy or with a medication and give people an edge on relapse. So our basic hypothesis was that if we could actually, in the lab, see this state, that we could actually use this as a way of screening our techniques for trying to be helpful in treatment. So what I'm going to do with this outline is glide us through in a very fast, not quite subliminal way, give you some examples of the way that we've actually approached capturing this ghost state and learning more about it and trying to link it up to our techniques for reducing it. First big question was, could we image it? I wouldn't be standing here if in fact we hadn't learned to do that. So we can grab this state, visualize it, capture it, objectify it in a laboratory setting, even though it's very sterile. So over here, it's just, we started out a really long time ago now in a pet lab with radioactively labeled water just as a flow tracer showing you where the action is. Doesn't show you the neurochemistry, just shows you where. And we could show that many of the little pieces of this motivational circuitry that have been shown in animal studies to be very important for drug motivation, for reinstating drug use in animals, could be demonstrated in humans. When we moved on a few years after that, with fMRI and our ever-expanding lab, Daniel and Terry, Marina, eventually Reagan, we basically were able to show, in fact, that across substances, not just for cocaine, but for opioids, for nicotine, for cannabis, and now Reagan's working with alcohol, you can actually see a lot of overlap in the areas activated when people see things that remind them of their preferred drug. We actually, as I said, had some collaborations where we're actually trying to look at the brain's response to natural rewards. We and lots of other people, because this became a very popular cottage industry to start looking at cue reactivity. So we've looked at and have published some areas regarding food cues, still ongoing, and sexual cues, which we think might be something that would give a window onto HIV risk, though maybe not in the way you would expect, and Paul will have one of these talks. He'll be able to tell you more about that. And the other thing that was interesting to me is that sometimes patients would say, I don't know what happened, I was just minding my business, I wasn't really going looking for anything, but I suddenly felt a pull to go look for drug. I was interested in whether or not motivation might begin outside awareness and that we could capture that with our fMRI paradigms, and we were able to by showing little teeny tiny cues, very brief visual cues, targets that we were interested in, and following them by things that were really boring. When you do that, you visually process the long stuff, but not the short ones. And we could show that, in fact, these little teeny tiny 33 millisecond cues were enough to get this system going and potentially get you in trouble. Are these things the same for everyone? If you've used cocaine for 20 years, is everybody equally reactive to the cues? They aren't. That was always the case. And we thought, well, maybe it's just because the cues that we're using have missed the mark a little bit for their conditioning history. But what we've come to appreciate is that, in fact, there's other sources of really interesting variability. So some of these are just good old genetic sources. As Terry was able to show several years ago now in the lab, a certain allele, a variant of the dopamine transporter, the little vacuums that suck up the dopamine that's potentially released in response to these cues, causing the trouble, actually had a very much bigger response to the cues. We were able to show recently, and interestingly, that a variant of the FKBP5 gene that's been known as a hypercortisol gene, it stars in studies of PTSD and depression and anxiety disorders. But interestingly, this variant also showed a really, really interesting association. People who had this hypercortisol, hyperaroused variant have a bigger response to these appetitive cues. So something that you think of as quote-unquote stress-related variant is boosting this. Interesting. A crosstalk between stress and reward systems that's important and that Paul will talk more about. He will when he gives his talk. Also, epigenetics, things that have happened to you. What if you've had bad things happen to you? Would that make you more or less reactive to drug use? So we had some very small questions in our screening materials, not really a proper screening, but where we asked people about bad things having happened to them, sort of as a prelude to a PTSD type screen. And we found that when people answered the bad things having happened to you trauma question, and you just split the data on this, and this is something that Jesse Sue did first, that the response to the little tiny cues was bigger in the people who'd had prior trauma. And Paul's published a wonderful paper in which he actually showed that people who have physical, emotional, or sexual abuse in their history have a bigger response to these cues. And Mike Ruziak, I don't think he's with us today, he's another collaborator, he's been with us for several years, showed that resting amygdala connectivity, amygdala being a front responder in these circuits, is actually higher in people who've had a prior history of trauma. Now, important, though these are all very beautiful brain pictures, do they mean anything in terms of the clinical outcomes of relapse, which I talked about at the beginning? And we have been able to show that, in fact, these cues, whether they are the 33 millisecond type, or as Paul's finishing up his manuscript now on the 500 millisecond type, or the 6-second type, people who are actually having a worse outcome, so whether they're having here, I should look, if I walk away, I can still walk away. Here, if they're actually having a rapid relapse, they have a bigger response in this motivational circuitry. Here, similarly. And over here, we divided people in a way that let us look at the worst of the worst outcomes. This group over here with their brain on fire, and the people who are going to proceed to better outcomes, about 30% of the urine's positive. That's considered a better outcome. And when we look at this, basically, across our different sorts of GO probes, we're able to show a link with relapse. So is there any hope with this? These things are pretty persistent. In fact, by their very nature, the brain response to these cues, and we see that from the days of polygraph work, is very extinction resistant, meaning that you can actually show these cues a lot without cocaine, and there's still a lot of reactivity. True for opioid cues, true for nicotine cues. This is the nature of the phenomena. So what can we do about that? Is there something we could do? Can we plop a medication on top of that? And if we did, what kind? So we do think there's some hope with this. We did, by the way, for years try passive cue exposure. That was tricky for the reasons I just described. You can sort of eventually get some reduction in reactivity, but you sort of pop back up like whack-a-mole. And we mentioned the collaboration with Michael Saladin, who's going to try reconsolidation. There's some interesting things that one can try, but a medication on board would give you some comfort that you were sort of calming this circuit down so that maybe the cues wouldn't have as much impact to begin with. And so what kind of circuitry? Well, we were able eventually, in collaboration with Nora Volkov's lab before she moved to NIDA, to show that, in fact, endogenous dopamine is likely released to these cues, as had been shown in animal studies with microdialysis and electrovoltammetry, but we were able to demonstrate it in humans. They gave us a good rationale for then asking about dopamine modulating medications. And we've had a couple of paths that we followed in our lab, and others will tell you about their particular pieces, I'll tell you about the couple that we've used for cocaine cues. So we've actually followed GABA B, and we followed this for nicotine as well. GABA B agonists like baclofen inhibit the cell firing at the level of the VTA, which is the origin of the dopamine cells that send their efferents out up from the brain through the striatum, through these motivational nodes. And what we were inspired by were Dave Roberts' work in the 90s showing that of the many, many medications he had screened for cocaine, he was looking hard with his preclinical models. This GABA B agonist seemed to show benefit. And it was encouraging to me because when they looked in animals, they could show that it did inhibit dopamine release that was in line with our hypotheses. And sure enough, we were able, with analyses of our brain response to the little tiny cues, to show that baclofen pre-treatment inhibited. You can see the red response is the brain response under placebo, and the blue response is the response under drug. And Ken Young is the author of that paper that was in J Neuroscience a few years ago. We also have been interested in whether or not we could get our hands, and I've been standing in line for this longer than I care to admit, for a D3 dopamine receptor specific, either antagonist or partial agonist. Again, based on preclinical literature suggesting that those should be really helpful. It's been really hard to get a specific one, but our stand-in is the medication Vraylar. You've seen it on television, Criprazine, House of Cards. I don't really like the ads, but it was approved a few years ago now, 3 I guess, for managing first mania and then depression in people who are bipolar. And it actually is an atypical antipsychotic, and when we have begun to try it, we have only a handful of imaging data at this point. We actually are seeing some encouraging bits that suggest that pretreatment with this D3 preferring atypical antipsychotic may be producing a reduction or a blunting in that activity. We don't know if it reads out as a clinical efficacy piece, but Kyle and I together have just finished a sort of a U54 mechanism where the idea was to get a lot of people that had measures of brain engagement and outcome and see if you could actually show that the brain response to the medication predicted the outcome. That is sort of the holy grail for us. So very quickly on this brief, almost subliminal presentation, we can image the brain response to these cues. There is individual variation. Genetics, epigenetics for sure, and I'm sure when Terry Reagan speaks to you, we'll talk about other modulators like sex and hormones. Can we link these things to relapse? Looks like we can. Is there hope with these medications? Yeah, going a lot more slowly than I would like, but yes. And what's on the future? What's on the horizon in terms of what we're doing now? So we have 3 activities that are ongoing. We have a newly funded P30 PET Center with Bob Mack and Hank Kranzler as the leads that will actually be giving us, hopefully, some PET tools that can be essentially juxtaposed with some of this medication development work. The focus of that center is, however, OUD. So if you have a good idea that is PET relevant and fits the ligands that are available here, we're going to put out a call for some new pilot projects for that center. So that's something that's very exciting. It's a new set of tools that we've lusted after, hoped for quite a long time. I'm also continuing with the R1 that I have to do the work with Criprazine and have the brain engagement measures and then follow it with outcome. And then Kyle and I are patiently awaiting, very patiently awaiting, not so patiently, it's been months, the Notice of Grant Award for a UG1 CLIN center that will allow us to measure target engagement to candidate medications and link it to outpatient performance. And we're going to do that in patients who are on opioid MAT of the naltrexone depot kind. But a lot of people who are on the naltrexone depot will sometimes use stimulants, which will then in turn keep them from coming back for their next injection. So we're going to use our anti-relapse medications on top naltrexone. And our candidates are the ones you see here. The commercially available ones would be Criprazine, cannabidiol, which is now approved for treatment-resistant epilepsy in children, and orexins. There's a dual orexin available right now, Balsamra, but we would like one that's orexin 1 specific if we can get it. And then we have ones that are not yet here. And I always have my hand out for these, but my hand has been out so long that it's going to cramp. positive allosteric modulators of the GABA B receptor would be a better bet than baclofen, better pharmacokinetics, better side effect profile, and we would love to be able to get our hands on a selective antagonist for the orexin 1 or for the dopamine D3. When we do this with the UG1, it'll be a paradigm that looks a lot like what we've been doing, where we have probes for reward, probes for inhibition, and we actually hope, as I said, to complement this, all with the goal of inserting the brain into this pipeline for developing medications to prevent relapse for cocaine, but also for the other substance use disorders. Everything I've told you should have relevance across categories, but I gave you examples mostly from cocaine. Thanks, guys. And of course, thanks to our funders.